Okay. Hi, good evening. Thank you for joining. Am I audible? And is my screen visible to you? Yeah. Okay. Thank you so much for confirming. Okay. Welcome to problem solving session 10. Uh, Today, we will be discussing the lectures for the 10th week of the course, which was about urea cycle and catabolism of amino acids uh, and the process which are part of the both of these broader concepts. So this will be 10th week. We have 2 more weeks to discuss the lecture, the syllabus. Like I mentioned in one of the previous session that I will be having one last session, which would be the 13th session. But however, unlike all the other course so far, the sessions that we have had on Tuesday 6 p.m., I am trying to put that up on 16th April, which will be the Wednesday after 15. 15 is where we have the 12th class. So on 16, we'll have that 2 hour session of the last session, which is basically a revision kind of a session. And I had mentioned last time that I'll put more of questions and solutions from this time's assignment as well, so that it's a good recap for you for whatever you are doing in this course. And I'm trying to put that on Wednesday 16. So hopefully that goes through, and we should get the confirmation, and I'll let you know in the next session if we would be having class on the 16th April. So after 15, the next class won't be on 22nd April. The last class will be on 16th April. That will be a problem solving session, which will be more focused on revising each concept that we have done so far. And I will be discussing the assignment questions as much as I can with the solutions and some important topics, or probably try to cover most of the topics with an example or a question from each, right? So yeah, that information I wanted to convey. And with that, let's begin today's class. So just a second. Yeah. So today's session, as I said, we'll be covering about nitrogen metabolism, and then we will be going to the second part, which where we'll be talking about amino acid catabolism, right? Okay, so let's begin. What is nitrogen metabolism, and where does it, what what purview does it fall under? So this topic which we are now discussing, which is nitrogen metabolism, this falls under the purview of biosynthesis of amino acids, right? So amino acids that we had done is one of the important biomolecules in our cell. And like all the biomolecules so far, we will be doing fatty acid in the next class, but we have looked into their synthesis, how they are produced, and how they are metabolized, how they are broken. So we are looking into the breaking and the making of each of these biomolecules. So today we will be focusing on amino acids, which are a component of the proteins that we know of, right? So in amino acids, one of the major components is nitrogen. So we will begin by first looking into the metabolism of nitrogen, because nitrogen, as we know, it ranks just behind carbon and hydrogen and oxygen. So it is also one of the major component which contributes to the mass of the living systems, right? So, but most of the nitrogen that you will see in amino acids are in nucleotides or amino acids, so they're in a bound form. So we will see how do you take nitrogen out of these bound forms, as well as how do you put nitrogen in this bound, how do you make sure that nitrogen combines with these molecules and becomes the important precursor for several pathways. And we will also see how do you take out nitrogen from these components and use it up and or remove it from the system if it's in excess. So we'll look into all of these different systems, right? Right. So the first thing that we will look into is the nitrogen cycle. Why is it important to first of all understand the nitrogen cycle? Why is it what are we seeing here? So as I already mentioned, the biosynthetic pathways that we know of that leads to amino acids and nucleosides, they share a requirement of nitrogen, because if you look at their structure, let's say amino acid. So what is the basic component? It has an amino terminal and a carboxyl terminal. The amino terminal has an NH2. So that N is important in amino acid. And if you look into nucleotides, they also consist of N. So nitrogen is a requirement for both the amino acids and nucleotides, right? But biologically useful nitrogen compounds are generally scarce in natural environment. You will not find them as readily available in soluble form. So in most organisms, they maintain a strict economy in the use of ammonia, amino acid, and nucleotides. All these three are components where you have the nitrogen, right? So we will see how the organisms are making use of nitrogen in different forms, and they are making use such that they can use it for synthesis of important compounds, right? So this one here, this figure here shows you the very important nitrogen cycle, how we are how are the different organisms using the nitrogen that is available in the environment for their own purpose, right? So which is the most important source of nitrogen? We all know it's air, right? And there are only, however, very few species that can convert the atmospheric nitrogen into useful form. So this one here, this nitrogen gas, as we know of, there are only very few animals or organisms that can fix this in a usable form. You do not directly have N2 that is being used as ammonia. So what is the majority driving factor? They are converted into these different forms that you see here: ammonium, nitrate, nitrite, and at different steps, you have different organisms that make use of that compound to convert it into a usable form. So in nitrogen cycle, what you need to remember is so mainly you should remember which are the components. That is very easy to understand. What are the three components? Nitrate, nitrite, and ammonia. Which microorganism is responsible for which conversion? That is what you need to take care of. So I will try to walk you through this figure, because as I know that you have already read this multiple times, but let's just do it once again, right? So in the biosphere, when these organisms are metabolizing, or when they are using the nitrogen for their own purpose, that is called nitrogen cycle. The first step of nitrogen cycle, where we have all read it so far, is nitrogen fixation. By fixation, I mean reduction, right? So the first step is nitrogen fixation of atmospheric nitrogen. Now that is done by nitrogen fixing bacteria. And what is the outcome of this? The outcome is ammonia. Just a second. Yeah. So some nitrogen fixing bacteria like Klebsiella, Azotobacter, right? These names you should remember. Please remember these names. These are the most commonly asked questions. So you should remember the nitrogen fixing bacteria, which is basically reducing the atmospheric nitrogen as ammonium or ammonia. Okay. So now, although these ammonia can be used by most living organisms, soil bacteria that derive their energy by oxidizing ammonia to nitrite and ultimately nitrate are so abundant and active that nearly all ammonia reaching the soil is oxidized to nitrogen or nitrate. So here you have this ammonia. As you can see, these are mostly used by as amino acid. They are mostly used by other components, but majority of them, this ammonium is used by nitrification or nitrifying bacteria, which are nitrosomonas. They convert it to nitrite. These nitrites again get nitrified by the soil bacteria Nitrobacter. Now this you need to be very clear. Ammonium to nitrite is nitrosomonas. Nitrite to nitrate is Nitrobacter. This is generally a very confusing part where people do mess up with the names. So please take note of it. That nitrate is then converted to ammonium by again reduction. Now the same ammonium that's being reduced from the nitrogen in the atmosphere, but not all organisms are able to do that. So majority, the remaining one, they do it by converting or reducing nitrate to ammonium. And that's done by some anaerobic bacteria and plants. Now what happens to this ammonium that is generated, that is used as degradation by animals of microorganisms to give we will see. This ammonium is what is used to make amino acid. And this is how from the amino acid also we will generate ammonium. And both this step of you making use of ammonium and liberating ammonium is what we will cover in today's class, right? So this talks about amino or nitrogen cycle. This is nitrification, and there is one more step that I had skipped, which is the conversion of nitrate to nitrogen. That's called denitrification. So whenever you're fixing nitrogen or whenever you're reducing it, it's nitrification or nitrogen fixation. When you are giving it back to the air, it's denitrification. Okay, so that's a simple balance, because all of these needs to take place in a balanced form so that it is optimum and there is equilibrium under all the conditions, right? Okay, now what you need to remember is who are these bacteria and where are they present, right? But before we do that, first let's understand the chemical nature of these reactions, or chemical nature of all these chemical, all the reactions that we have just read, right? Now we know that only certain bacteria can fix atmospheric nitrogens, and as I mentioned, these have azotobacter or the Klebsiella that we had just read. And there are also some nitrogen fixing bacteria that live as symbiont in the root nodules of leguminous plants. So leguminous plants have some nitrogen fixing bacteria which exist in symbiosis with them, right? Now we will look into where these bacteria are present, but before that, we will see how does this change take place and why does this conversion doesn't take place so easily, right? Now if you look into this, nitrogen to ammonia, which is this reaction here basically, this is highly exergonic. This has very high delta G, which is a negative. So it's supposed to be a reaction that is exergonic and that should take place very easily. However, that doesn't happen, and that doesn't happen because of this here. This nitrogen, if you know, just a second, if you know nitrogen, they are bond like this to each other. N2 is basically this bond. Now this triple bond is what makes it a problem. This triple bond makes nitrogen very, very stable. And this bond has a bond energy of 930 kilojoule per mole. Look at the delta G for the overall reaction. That's minus 33.5 kilojoule. But just the bond which is between the two ends, which is the bond that's holding these two nitrogen, that's 930 kilojoule per mole. Therefore, nitrogen fixation becomes extremely, like it's not really in a spontaneous manner because it requires very high activation energy. And that's why atmospheric nitrogen is chemically inert because of this high bond energy. For you to make use of that nitrogen, you need to break this up. Now, in industry, you would have read already that there is something called Haber's process, which makes use of energy and all this for breaking the bonds. That's not happening here. That cannot happen in biological systems. So how do the biological systems or organism make use? Where do they get this activation energy from, right? So in biological setup, nitrogen fixation occurs. So as we know that ATP hydrolysis, ATP binding, and we know it's binding and hydrolysis is not just hydrolysis. We have established this already, that when you are using ATP as an energy currency, it doesn't simply just bind or so doesn't simply hydrolyze. Both the methods of binding, forming an intermediate, and hydrolyzing is what is giving the energy. So that ATP is driving the high activation barrier here, and that makes use of the hydrolyzing energy of ATP to finally give you ammonium for nitrogen. So so what I meant to say in overall here is that when you have a conversion of nitrogen to ammonia, you don't simply have this reaction, even though it looks like this. You need to use ATP. So in the biological systems, you have ATP, which is used to drive the conversion of nitrogen to ammonia, along with some H plus and electrons. That's basically reduction, right? So overall, if you look, and you should remember this stoichiometry as I have always mentioned. So you have N2 plus 10 H plus, 8 electrons and 16 ATP, which gives you 2 ammonium molecule and 16 ADP and inorganic phosphate and 1 H2, right? So this is important that you should remember. Now, what is the enzyme that does this? There has to be an enzyme definitely. If someone is making use of ATP, that entity is an enzyme, and the enzyme here is nitrogen complex. This nitrogenase complex consists of two enzymes as this component, which is dinitrogenase reductase and dinitrogenase. These are the two enzymes that are responsible for carrying out this conversion and by making use of ATP. Right now, this dinitrogen reductase and dinitrogen, if you look into the notes, you will see that each of them have their own structural component. One is with 4 FES center, one has homodimer, but both of these, what is important for you to remember is both of these have redox centers. Both of these has, if you remember, iron sulfate centers, and they make use of these centers for catalyzing the reduction that we look into here. Right now, what you need to, what I will be now focusing is how is the stoichiometry taking place. Right, so this nitrogen fixation, as you can see here, and as we have already established, it is carried out by this highly reduced form of dinitrogenase and requires 8 electrons. So where is this highly reduced form of dinitrogenase? So if you see something very interesting here, for this panel conversion also, so let's go back first. Instead of me starting from the beginning, you have this nitrogen which is converted to ammonia. Now, while this reaction is taking place, nitrogenase is getting oxidized. Of course, because if nitrogenase is getting oxidized, then something else has to get reduced, and that reduction is of nitrogen. So dinitrogen getting oxidized, and in the parallel, you have nitrogen becoming ammonia. This step requires 8 electrons. Okay. Now, where does these 8 electrons come from? It's not just directly from it. It's also a reduced form. That means it exists in both oxidized and reduced form. So who is reducing it? Reduction is being taken. Reducing the dinitrogenase is dinitrogenase reductase, which here is where you have 16 ATP usage. Right? So this uses 16 ATP, and this reduces dinitrogenase. Now, this is reduced again. Where it is getting reduced from? This is being reduced by ferredoxin. And where is ferredoxin receiving its electron from? We go back to the same components that we have been studying since the beginning. Coenzyme A and pyruvate. Where were these components? These are components from glycolysis. So when you have coenzyme A and pyruvate, which is converting to acetyl coenzyme A, this is a defined step of glycolysis. The electrons that are generated here go to ferredoxin, reduce them. These ferredoxins get oxidized back, and while getting oxidized, give that electron to dinitrogenase, which gets reduced, uses 16 ATP, and in the reduced form, then it reduces. While it is getting oxidized, it reduces dinitrogenase, and that gets oxidized. So you can see why is it called a complex? Because it has several components that take part together for the final reaction. And this is the 8 electrons that have moved from coenzyme A to ferredoxin. That is being transported in this cycle. And in this step, it also uses 16 ATP. What you need to focus and what you need to remember here is, in the nitrogen complex, what are the two components and what are their order? Which one is coming first and which one is coming later? And which is the starting point? So if you just remember in that way, it should be easy. And the name suggests, right? You need to remember that it starts from ferredoxin. It goes to dinitrogen reductase. The name says it's a reductase enzyme. Whom does it reduce? It reduces dinitrogenase. So the dinitrogenase comes later, and dinitrogenase is for, as the name dinitrogen suggests, it is 2 nitrogen. So nitrogen gets converted to ammonia. Right? That's our entire gist here. Now, well, that's clear to us that nitrogen fixation is happening by what mechanism? And we know who is also converting otherwise nitrogen. All of these are clear. Now we'll come to the symbiotic bacteria which are present in the root nodules of leguminous plant. And why are they fascinating? They are fascinating because of something which is called leg hemoglobin. But before understanding leg hemoglobin, we will understand one important concept again here, or one important thing that is to be remembered here is this. So nitrogenase complex that we have just looked into the previous slide, it is an extreme lability in the presence of oxygen. So the reductase, it gets inactivated in air. And what does oxygen do? It gets oxygenated or it gets oxidized. That's why This is why I didn't mention in the slide, but I will mention it now. Whenever you are in the oxidized form, these entire complex is inactive. So this doesn't take place. As soon as it gets oxidized, it becomes inactive, right? So it needs to be reduced, right? So this reductase that we have just looked into is inactivated in air. Now, free living bacteria that fix nitrogen, they have variety of ways of coping with this. So some bacteria which are anaerobic, they are able to carry out this entire reaction, right? Because they're anaerobic, they don't require oxygen, they will never come in contact with oxygen. But some aerobic species such as Azotobacter, what they do is they uncouple the electron transfer from the ATP synthesis so that oxygen is not used. So you're partitioning the two ways such that you are never encountering oxygen, right? Now, these organisms, when they're fixing nitrogen, these culture themselves maintain a, what do you say, checkpoint to reduce oxygen. So these bacteria, the ones that are aerobic and they need to make use of nitrogen complex, nitrogenous complex, they make sure that they are doing it without the use of oxygen. From this concept is what is important for you to understand leg hemoglobin. Leg hemoglobin, the name suggests where are they present. Leg is basically leguminous. So this leg is leguminous and a hemoglobin. Now what does this hemoglobin do? So this hemoglobin or this leg hemoglobin, it's a solution that these bacteria which are present in the root nodules of plants have come up with for controlling the oxygen toxicity problem. So these bacteria, they are present in this solution of leg hemoglobin. This is an oxygen binding heme, which is produced by the plant. So this is produced by a plant. So plant gives this hemoglobin. This hemoglobin captures all the oxygen. Now, because this binds to all the available oxygen, this oxygen cannot interfere with the nitrogen fixation. And therefore, because you don't have oxygen, the bacteria is easily capable of fixing all the nitrogen. So this is important in symbionts. And what is the symbiotic association here? The symbiotic association is that leg hemoglobin is given by the plant, and that is utilized by these bacteria which are present in the root nodules. And what is the protein called? Leg hemoglobin, right? These key terms you need to remember. Okay, all right. So we now know how bacteria fix. We now know what is a symbiotic nitrogen fixing bacteria doing, how are they present, and what is the entire process that we have just looked into. Now let's look into a bit more detail. We know this overall reaction. Now let's look into this step by step, what's exactly happening. Okay. Now, just a second. Yeah. Now, after the nitrogen is reduced, what's exactly happening? So this reduced nitrogen that we have just come into, NH4, how is it being used? So this reduced nitrogen, which is in the form of NH4 that we have already established how, this is assimilated. Where did we study assimilated previously? We read in carbon assimilation. What did we know about carbon assimilation? Is basically how are carbon used to produce organic compounds. We have learned breaking, we have now looking into making. So now we are see how nitrogen is being made or assimilated into the compound that is its part of, which is amino acid and dinucleotides. So the reduced nitrogen is assimilated into amino acids and then into other nitrogen containing biomolecules. And that is mainly done by these two amino acids, glutamate and glutamine. I told you we'll be doing two things today. We will be looking into the amino acid making and amino acid breaking. When we go to amino acid breaking also, we will encounter these two amino acid, glutamate and glutamine. So you need to be very clear when you are looking into these topics that where exactly are the enzymes taking place. Enzymes, as you will see, we have glutamine synthase. You will find many other enzymes in the next section. So you need to be clear in your mind what you are doing. Be very careful when you are studying these topics. Which enzyme is part of which pathway, of which metabolism. Okay. Now let's now look into the ammonia assimilation. How is it being assimilated, right? So the two amino acid, as I have said, glutamate and glutamine, they provide the critical entry point. Okay. Among this also, glutamate is the source of amino group. Now, what are these amino acids important for? They are important for the amino group again. And these, among these also, glutamate is a source of amino group for most other amino acid. And this is by transamination reaction. Where does it look? Transamination. Oh, this on the other side. Okay, so via transamination, which we will see in the next part, by those reactions, you have the glutamate, which is giving the amino group, right? So the amine nitrogen in the of glutamine is a source of amino group, again in a wide range of biosynthetic process. Okay. Now here, what happens is the glutamine synthase, as you can see here, this catalyzes the reaction of glutamate and NH4 to yield glutamine in two steps. However, what is the first step? In the first step, you have glutamate, which binds to ATP, and then there is this intermediate here, which is gamma glutamyl phosphate. So there is a phosphate that has transferred from ATP to glutamate. So it becomes glutamyl phosphate to ADP. In the next step, the ammonium, which is supposed to be used now, it gets incorporated. The glutamate with ammonium in it becomes glutamine. What is given out is the inorganic phosphate. So overall, when you see, you have glutamate, which made use of ammonium, which made use of ATP to give glutamine, ADP, PI. What is important for you to remember is this step here. This is not directly transfer of amine. There is an intermediate here, and that gets converted to glutamine by exchange of phosphate with ammonium. This enzyme, which facilitates this, is called glutamine synthetase. Very, very simple to understand. Glutamine synthetase. Tase means use of ATP. There is something called synthase. There is something called synthetase. Wherever you find the term synthetase, tase means something that has used ATP for synthesizing something. What is the something here? Glutamine. So glutamine synthesizing enzyme using ATP. ATP and glutamine synthase. That's how you remember these two enzymes, synthase and synthetase. Okay, now that's one. What is the other enzyme? The other enzyme is glutamate synthase. Now you have glutamine here, you have alpha ketoglutarate, which is there, and that's use of plus the NADPH, which gives you two molecules of glutamate and NADP. So what you are seeing is the same glutamate that was used in the previous step has been regenerated here, right? Now, where is this alpha ketoglutarate coming from? This we already know, it's an intermediate of Krebs cycle or citric acid cycle. And here it goes something called reductive amination. Now, there are 2-3 names that I am constantly using: transamination, reductive amination. Again, because it's of importance here, you need to know the distinction between them. You need to know which one is what. Glutamate synthase goes of the enzyme, the reaction catalyzed by glutamate synthase is reductive amination. Okay, so you need to know the name, like where is transamination and where is reductive amination. Right now, so you have glutamate back. Now, if you overall combine these first and second reaction, you get alpha ketoglutarate plus NH4 plus NADPH plus ATP gives you glutamate, L-glutamate, NADP, ADP. So where did the glutamine go? So if you look into the left side hand side of these 22.1 and 22.2 reactions, glutamate and glutamine are present in the reactants. If you look into the right side, which is the product, you again have glutamine and glutamate. So these get cancelled out. So what remains are the other components which are not common to both these reactions. So you have alpha ketoglutarate plus NH4 plus ATP, NADP, H, and that's being used, and that gives you glutamate and ADP. So what it means here is that when you have your ammonium, which is from the reduction of nitrogen, how is that ammonium incorporated? Is by it's brought into the cycle by this glutamate, and from this glutamate is where you have the L-glutamate, which is further carrying these out in the other biomolecules. Okay. Now, Okay, now let's look into this. So we've already established this enzyme. Okay, I forgot to mention glutamate synthase, which is not difficult to understand because it is generating glutamate. So here is this net reaction. However, this enzyme is also there. L-glutamate dehydrogenase. If you look into this reaction and this reaction, it's basically the same. So what is happening with this enzyme here? What is this enzyme doing? So the previous enzyme, glutamate synthase, this is not present in animals. Okay, which so in animals you don't find glutamate synthase, and therefore in these animals they maintain high level of glutamate by processes such as transamination of ketoglutarate during amino acid catabolism. So in these animals which do not have glutamate synthase, they do transamination of ketoglutarate. We will see this in the next part. However, there is another step that can take place where alpha ketoglutarate, which is from the citric acid cycle, it forms directly glutamate in a single step, which is catalyzed by this L-glutamate dehydrogenase. So it's directly saying that it's L-glutamate dehydrogenase. So instead of both this enzyme, it's only in bacteria and other organisms it is present, but in animals this enzyme is not present. So how do you come to this L-glutamate? You can directly go from ketoglutarate to L-glutamate by using glutamate dehydrogenase, or you have transamination of alpha ketoglutarate. I know it sounds very confusing when I am talking in one single manner, but what I am trying to highlight is there are several alternate routes for the similar kind of reaction. But what you need to understand is all organisms make use of similar machinery to come to the same compounds, because same compounds or similar compounds drive the entire biosynthetic pathways. Okay, so you need to come to L-glutamate, you need to make L-glutamate. If you won't have one enzyme, make use of other enzyme, but the ultimate result or ultimate compound has to be L-glutamate, and that is what is important for us to understand, that your L-glutamate is the final product that is utilized for production of different metabolites. Okay, right. Now lastly, as always, as we know that whichever enzyme is crucial for the entire pathway, we have already looked into this glycolysis, we know for gluconeogenesis that whichever enzyme is most crucial or whichever step is most crucial, that step is highly regulated. So this is the most important step here. Glutamine synthetase, that's the first step that incorporates the ammonium generated. So what happens if you do ammonium generation? You have production of amino acid, you have production of nucleotides. That means, think in this manner, you are producing amino acids, you are producing different amino acids, making the concentration higher. You are regulating the entry of, or you are regulating the production of amino acid by blocking the first step, which is glutamine synthetase, and that's why that is highly regulated, right? As you can see in this image here, that glutamine synthesis is regulated allosterically. So it undergoes cumulative regulation by 6 end products of glutamine metabolism, right? And alanine and glycine serve as indicator of general status of amino acid metabolism in the cell. What does it mean? Let's look at the crosses. I told earlier, crosses means they regulate in negative way. How? If glycine is present, glycine will inhibit this enzyme. Why? Because glycine is an amino acid. It is already present. It means there is no more need of amino acid to be there. It will stop the production. So it will stop glutamine synthetase by allosterically inhibiting it. Similar to it is alanine. Both of these are amino acid. Now, other than this, you have these 1, 2, 3, 4, 5, 6 intermediates of the pathway itself. We have already understood. This is not a difficult concept to understand. You should just remember that whatever is the product, that can only inhibit the step if it is higher in number. For example, AMP. What is AMP? AMP is based when you have glutamine breaking further. Glutamine gives rise to AMP, tryptophan, carbamoyl phosphate. We will encounter this in a while. glucosamine, histidine, CPD, any city, all of these compounds, if they get higher in concentration, they will inhibit this step itself. It will, so it's called negative feedback. It, when the concentration of the product is higher, it stops the pathway that leads to its creation. So this is one way, which is simple to understand, that its own intermediates will inhibit its formation or inhibit the pathway. What is the other? The other is when you have covalent modification. What is covalent modification? Your enzyme is modified by covalently attaching with a adenylated tyrosine. Adenylated tyrosine. What does it mean? It means that uh, there is an adenylation. It's basically addition of AMP or here. You can see it here in the slide, this adenine. So adenine monophosphate. So you have an adenine monophosphate that is present, or that is bound to the tyrosine 397 at the enzyme's active site. So basically, this is your enzyme. This is your tyrosine. And in this tyrosine, this is tyrosine, you remember? And in this tyrosine, you have this adenine monophosphate, AMP, adenine monophosphate binding. And what does it do? It makes the enzyme inactive. So this covalently modifying these enzyme increases the sensitivity to allosteric inhibitors. Basically, if you have these modification, it's more sensitive to bind to the allosteric inhibitors, all the inhibitors that we just saw in the previous slide. And as a result, it gets regulated. And who adds this adenyl group onto the enzyme? Very easy, adenylyl transferase, right? So if you have adenylated enzyme, which you will see here, AT is basically adenylated. So if you add adenyl on the enzyme, this enzyme becomes more sensitive to all these compounds, and therefore you are regulated, right? That makes sense. But what is this UT here? Why is there an UT here, right? So you need to understand what is this UT. Now, UT is basically uridylyl transferase. What is that doing? So this adenylated or adenylyl transferase, the activity of that enzyme is modulated by binding to a regulatory protein called P2. And the activity of P2 in turn regulated by covalent modification, again at a tyrosine residue, which is basically adding of uridyl or uridyl residue. So the adenylated transferase complex with uridylated P2 stimulates the deadenylation, whereas the same complex with deuridylated P2 stimulates adenylation. So there is this close, how do you say, balance or close equilibrium between uridylation and adenylation. They both go hand in hand so that the overall regulation takes place. Let's look into this flowchart to understand what's exactly happening. So this is the reaction. Alpha ketoglutarate getting converted to glutamate, and that's finally getting converted to glutamine. That is what exactly the reactions is all about. Glutamate to glutamine. Glutamate to glutamine. Right? Now, which enzyme is taking place here? Glutamine synthetase. This glutamine synthetase is active, this reaction takes place. Now, when glutamine synthetase is not active, it's basically when it becomes adenylated. Who adenylates this? Here, AT adenylates this. But how does AT does that? When it binds to P2. But when will AT bind to P2? Yes, it's complicated. That's why I am walking you through. If ever you get confused, you can walk it in this way. I will do it 1 second. Let me just complete one cycle. So your AT, A means adenyl. Adenyl is for glutamine synthetase. Okay. So AT, when it binds to P2, then it can do this, or that it can catalyze this reaction. But when will AT bind to P2? When P2 itself is bound by UMP, or when P2 is uridylated, and that uridylated comes from so this is P2. This P2 is basically with when it is in a P2UMP form, and that P2UMP form with ATP, if it is bound to AT, it will make it active. Okay, now you see what's exactly happening. If it is bound to AT and UMP both AT and UMP, it makes it active. Okay, but if it's only bound to AT without UMP, it gets inactive. So if there is no UMP, it can bind to glutamine, and so the adenylyl can bind to glutamine and it can become inactive. If it is bound to UMP, it is active, means it cannot bind to the adenylation. So you need to, if you are here and you want to inactivate, you remove the UMP. So this UMP is removed, your P2 is free, it binds to AT and it then binds to adenyl. Okay, it's a bit complicated, but once you get the flow clear, if you have both of them, you have AUMP and AT, then what it does is it activates the glutamine synthesis. If it only has AT, as we know, adenylyl transferase does adenylyl transferring. But adenylyl cannot transfer the group if it is bound to UMP. It's as simple as that. Okay, so that's how it's regulated. And this is the first checkpoint, because it's the first step which is crucial for the assimilation of nitrogen. Right? It's complicated if you look into the first time, but once you understand the logic behind it, it becomes easy. But what you need to do when you are revising is you need to take care of the names. The names in biochemistry mostly give away what kind of process it is involved in. Adenylation, synthase. Few name you need to remember, and that should make your life easy. Okay, so in the first part today, what we looked into was how nitrogen is fixed, what is the final product, and who fixes it, in what process, and what happens to a special case when you have leg hemoglobin. And ultimately, when you have the ammonium, how is it made use to produce the amino acid, which is the L-glutamate, which is the beginning of all the other amino acids, right? And then we looked into regulation as always. So that's the first part of this week's lecture. Now, what is the second part? This was making. How do we break it? How do you catabolize? Sorry. How do you? Yeah. How do you catalyze these amino acids? What is the catabolism steps? So amino acids get oxidized, and you have concomitant production of urea, and that's your process of breaking the amino acids. And that's what we are going to do now. Okay, so amino acids are basically oxidized in cells, unlike, so we did the opposite, right? When we were looking into the production of amino acid, we were looking into oxidation steps, right? But here we will look into the we will look into reduction step. Here we will be looking into the oxidation steps, right? Now, how does amino acid get oxidized? These are the three methods that can undergo, that cells can undergo. So here you can see. The first thing is, if there's a normal synthesis and degradation of cellular proteins, some of the amino acids that are released from the protein is basically amino acids together. You are breaking, fragmenting the proteins. Some of the amino acids will come out. These amino acids, which are not needed for any other, so you have the monomeric units of amino acid, and you don't need the amino acids for building a more protein. It undergoes oxidative degradation. That's one. What else do you see? So if you are eating something which is rich in protein, and your amino acids which are ingested are high, the requirement has been met, and still there is excess remaining, then your supplies is catabolized as amino acids cannot be stored. So amino acids are not stored. Fatty acids can be stored. We will see. We know glucose can be stored, but amino acids cannot be stored. So amino acids need to be excreted and need to be removed, because amino acids they are needed for the production of protein. If you don't have the need for generating protein, or if you don't have the need for building any more protein, then the amino acids are removed. And we will look into that how. Also, when you are starvation, when you are fasting, when there is starvation, or there is uncontrolled diabetes, that time, and when the carbohydrates are not available, that time you are using proteins as fuels. So proteins can be used as fuels, and amino acids cannot be stored. So in all these steps, your amino acids oxidation has to be done. And in oxidation, we mean that it is basically losing its native structure. So what happens is your amino acid, they lose their amino group. See, again, the amino group is what if you remove from amino acid, it is degraded. So they lose their amino group to form alpha keto acid. Basically, if you remove the NH2, it will remain as CO, alpha keto acid. And it's basically just carbons. So it's also called the carbon skeleton of amino acid. Alpha keto, so any amino acid, say glutamine for that matter. So if you, from glutamine, if you remove N, it will become alpha keto acid of glutamine. Okay, so the alpha keto acid is something that is the carbon skeleton that remains after an amino acid loses its amino group. So alpha keto acid, it undergoes then oxidation to give you carbon dioxide and water. Or if that's not the case, it will break down further and give you more 3-4 carbon unit, which is utilized for other purposes. So you see, even the carbon skeleton is used for different reasons. So this is how summarizing that. If you have a protein, or if you have there's an intracellular protein, or if you have ingested protein, in both of these protein you have amino acid constituents. The how is the amino acid broken? First part is NH4, which is amino group, and the other part is carbon skeleton. Carbon skeleton is basically alpha keto acids. They're called the alpha keto acids, and they enter citric acid cycle to give CO2. We will see why. All the names, if you remember, succinate, fumarate, arginose, we will see fumarate, malate, not malate exactly, but fumarate. All of these are formed from one or the other amino acid enter the citric acid cycle. So carbon skeletons are easy to take care of. And from there, we have already looked into when you have the oxalic acetate that goes out and takes part in gluconeogenesis, right? What happens to ammonium? This ammonium here is used in the biosynthesis of amino acid nucleotide. This is something we have just discussed. If this ammonium is taken by glutamine synthase, it's called amino acid production, right? But if that's not the case, then what is it doing? It has to be out of the cell. So it forms something called carbamoyl phosphate. We will see that enters something called urea cycle, and the urea is excreted. And from here, between these two, you have the aspartate arginosuccinate shunt of citric acid cycle that what do you say, that equilibrates the concentration here among these two. So that connects the urea cycle, that is throwing the urea out, and it connects the citric acid cycle, right? So you have these two byproducts of amino acid catabolism. Okay, now let's look into both of these individually. We'll not look into carbon skeleton as much, but we'll look into the ammonium first. So here you can see. So this degradation of amino acid takes place in liver. Don't forget the site. The degradation takes place in hepatocytes. Hepato means liver. So hepatocytes means liver cells. So the degradation takes in liver cells. So you have a cellular protein, or you have ingested protein, whichever protein you have, that's giving you the amino acid. This amino acid that we saw just now, this amino acid will give out nitrogen or ammonium, and in turn it will become alpha keto acid. Let's look into this. This is COO, this is CH, this is R, this is NH3. When it loses the ammonium, it becomes, it occupies this conformation where it has a keto group here. That's why it's called alpha keto acid. If you want to look into it into more detail, it looks like this. So what happens? Who takes the ammonium there? Again, here you have alpha ketoglutarate, again alpha keto acid, which takes this ammonium, which is being removed. It takes ammonium and it is adding itself on the alpha carbon, and it now becomes glutamate. So reverse is, if glutamate loses ammonium, it becomes alpha ketoglutarate. See, the name also suggests what keto. So alpha keto is this one. The next part, glutarate, is basically the amino acid that it has come from. So alpha ketoglutamate, glutarate has come from glutamate. So if you remove ammonium, it becomes alpha keto. If you add ammonium to alpha keto, it becomes the amino acid, right? So this has taken place while you have a alpha ketoglutarate. Now this amino acid can be anything, and we will see biglutarate or alpha ketoglutarate here, right? So you have glutamate synthesis, glutamate that has generated. Here this glutamate will again become alpha ketoglutarate, but while it becomes so, as we know, it will lose this ammonium. This will go out of the liver cells. Okay, so that's one. Also, simultaneously, you will have glutamine that we have already established. This glutamine itself can convert to glutamate, and it can also give ammonium. This we have also established. There is this very important and very interesting case here, which is alanine from muscle. We will see this. That can also become glutamate. Okay, while glutamate, and then again you have a glutamate that will become alpha ketoglutarate. So, what you need to remember? One amino acid is there, let's say any amino acid, it needs to give out its ammonium. Who will take the ammonium? Always the alpha ketoglutarate. We will see why. So alpha ketoglutarate will see, okay, amino acid is there, I will go take the ammonium. When ammonium is attached onto the alpha glutarate, it will become glutamate. So that means glutamate is very important amino acid. It is the one, it is the enzyme, it is the amino acid that is responsible for excrete ammonium as well as making use of ammonium. So glutamate will take ammonium, and then in the same reaction when it has to give the ammonium back, it will become alpha ketoglutarate gives away the ammonium, right? This glutamate is what is important for getting the ammonium out of the cell. Now glutamate can be formed by glutamine. Glutamate can also form from alanine. In both the form, it will give out ammonia, which will then be excreted by the cell, right? How are the ammonium excreted from a cell? We have seen this multiple times in our literature. We know ammonium can be excreted as ammonium, ammonium can be excreted as urea, or ammonium can be excreted as uric acid. Where do humans form? Humans fall here. We excrete urea. Animals which secrete or excrete ammonia in their when they don't modify ammonium any further, they are called ammonotelic animals. Ammono means ammonia. So they are excreting ammonia. And these you will see in fishes. Uric acids you will find in birds and reptiles. Urea you will find in all the terrestrial vertebrates. They all secrete urea, or they all excrete urea. Okay. And we also come under urea. We are called the ureotelic, while the ones which excrete uric acid are called uricotelic. Very simple name, not at all confusing if you look into the logic. Okay, so we have understood the overall reaction that is taking place. Now let's dig deeper and see what's exactly happening. And this is where this name comes again. We have looked into reductive amination. Now we will see transamination. Let's look into this without getting confused. Again, let's start here. Alpha ketoglutarate taking ammonia from any amino acid that is undergoing oxidation in liver. So alpha ketoglutarate takes the ammonia from an L amino acid, it becomes L-glutamate, and this amino acid forms an alpha keto acid. Easy to understand. This reaction is called transamination reaction. That's also easy to understand. And to understand in more how do you say technical terms, it basically is the alpha amino group here is transferred to the alpha carbon of the ketoglutarate, leaving behind an alpha keto acid, which is analog of the amino acid. Okay. But additionally, you need to remember is this component here, PLP, which is called the pyridoxal phosphate. This pyridoxal phosphate is important for this catalysis that is just mentioned. So when you have transaminases reaction, transamination is also called amino transfer, or aminotransferases are also called transaminases, right? So these both are the same term. Now these aminotransferases, they have this prosthetic group called PLP, as you can see on the slide, which is called pyridoxal phosphate. This coenzyme form is of pyridoxine form, or it is from vitamin B6. So this pyridoxal phosphate, it functions as an intermediate carrier of amino group. So it carries the amino group on its active site, of the amino transferases, and then it undergoes some reversible transformation between several of its forms like aldehyde, pyridoxal, and several other forms, and it finally gives the amino group to the alpha keto acid. So it basically the amino group that is transferred from the amino acid to the keto group is basically carrying the amino group. So the pyridoxal phosphate, the prosthetic group, is carrying the amino group. Okay. Now this pyridoxal phosphate is generally covalently bound to the enzymes. So that's why it's a prosthetic group, as we know, right? So it is bound covalently to the enzyme's active site through an albumin Schiffs base. So this is how it looks. Okay, this is a Schiffs base here. Okay, so this is your pyridoxal phosphate in general. This is your pyridoxal phosphate with the amino group when it is transferring. It becomes something called pyridoxamine phosphate. Or when you look into the enzyme overall, this is the enzyme. To its lysine, you have these group bound here. Okay, so this is carrying amino group. It binds to the lysine group, and it forms something called Schiffs base. And this is how this ammonium is transferred. Where is it taking place? Just for you to remember, here you have the amino acid. Oh, sorry, you have the amino acid. This amino group is transferred here. Okay, and this amino group is transferred here, and it is then giving it off to the alpha ketoglutarate. While this enzyme and this group are together covalently bound, and this is how they are bound. So enzyme's lysine group, amino group, is bound to the PLP, and this forms something called Schiffs base, something like this. Okay, you would have seen this in the notes as well. So if you look carefully into this, it's basically showing you the amino acid transformation of alpha carbon, which is facilitated by pyridoxal phosphate. So if you look into this, the PLP is bound to the enzyme through a Schiffs base, right? Now, if you see at the reaction at the top left, this is where the reaction begins with the formation of a new Schiffs base. So Schiffs base, you will have words like this also in the question. Schiffs base is called aldimine. Aldimine means aldomine, imine group. So mine means nitrogen. So aldimine group between the alpha amino group of the amino acid and PLP, right? So this Schiffs base that you see here, this guy here, Schiffs base, this has 3 intermediate. So one is it can take the quinoid form and then finally give the pyridoxal phosphate back and the alpha keto acid, or it can again become quinoid intermediate and it can give pyridoxal. See, everywhere it has the pyridoxal form comes out, right? But the subsequent product is different. Here you have the alpha keto acid, that is what we are concerned with. But it can also give you D-amino acid. It can also give an amine. So this Schiffs base doesn't only do this reaction, it can also give you different other reactions. Okay. But what we are more concerned with amino acid catabolism is this one. This takes place in the hepatocyte. Hepatocyte is where you have the amino acid catabolism or amino acid oxidase, right? This reaction is called transamination. The second reaction, B, is called racemization, and third is called decarboxylation. You don't need to remember all of these, but what you need to remember is that the PLP can undergo several reactions, out of which when it undergoes transamination, that is what is taking part in amino acid oxidation. Okay, so when you see this image, just remember that these are the different kind of traits that PLP can have when it's bound to the enzyme. Okay, out of which transamination is what is utilized for amino acid catabolism. Okay, all right. Now, so this is what I meant. You have transamination where you have alpha ketoglutarate that is being transported. Oh, sorry, that is being that is being converted to L-glutamate, and subsequently you have a alpha keto acid from the respective amino acid. This reaction is called transamination. Okay, this one's clear. Okay, now what happens after this? What happens after all of this thing has happened? Now ammonia is there. Remember, why are we even throwing ammonia out? Why can't we just keep ammonia, because we need ammonia for production of protein? So if ammonia is there stored in some cells, then it can be utilized later. That is what we are doing anyway with everything else, right? But why can't we store ammonia? Because ammonia is toxic. So ammonia cannot be stored because it's very toxic to animal tissues, and that's why whenever it is excreted out of hepatocyte, it needs to go away. It needs to directly excrete, right? So you will more often than not see that the level of ammonia which is present in the blood is often regulated. So in tissues like brain, they have uh, nucleotide degradation, which directly generates free ammonia, right? Now, in these kind of cases where ammonia is converted, it is to be converted to a non-toxic compound before it is being brought to liver, right? Somewhere else. So mostly ammonia is produced in the liver when your amino acid is entering, right? But you also have in your cells different places where directly ammonia is synthesized. Now imagine you have ammonia in somewhere in brain that has to be transported to your liver. While it is traveling in the blood, it's not safe because it's toxic. So what it does, it is converted into another form before it is brought to the liver, transported to liver or kidney, wherever. So what happens here for the transport function? Again, glutamate is important, right? So your glutamate, which is critical to the amino acid metabolism, it is supplanted by glutamine. So the free amino acid which is produced in these tissue, they combine with glutamate to produce glutamine. Again, we are doing the same song and dance that we have been doing so far. We have absolutely like no shying away from the fact that glutamate is very, very important for our cell metabolism. So this here, when you see, is basically you have the ammonia which is coming in contact with glutamate, gets converted to glutamine, and then in glutamine it is transported through the blood. It's brought to hepatocyte. The first step in hepatocyte for glutamine will be L-glutamate. When you have ammonia relief, your ammonia is relieved, it becomes N-glutamate. So the ammonia which is in the blood, just remember it like this. Outside hepatic tissue, if you are ammonia, this ammonia also binds to glutamate and becomes glutamine. The reaction that we have seen in the previous part where we were looking into how ammonium is being utilized. So glutamate uses ammonium to become glutamine, and glutamine is transported, right? So that's about how is this taking place. Now, what is the step here that I have left? What is in center? In center, you have something called oxidative deamination. So what is happening here? So in this, in the liver cells, the glutamate is transported from the cytosol into mitochondria, and the glutamate undergoes oxidative deamination by this enzyme to give your NH4. So when you have glutamate is deaminated by glutamate dehydrogenase, right? Now I was talking about why do we why do we always need the what do you say, alpha ketoglutarate for the step here, aminotransferase. That's because this enzyme is more is particularly specific to alpha ketoglutarate. So whenever this aminotransferase, amino can be any amino for that matter. It can be alanine transferase, it can be alanine aminotransferase, it can be any aminotransferase. For that aminotransferase, the substrate is always ketoglutarate. No matter which amino acid it is, it will always work specifically for alpha ketoglutarate. That's why you will always have alpha ketoglutarate at its beginning. While when you have this glutamate, which is this, for it to become alpha ketoglutarate, but which enzyme takes place mainly is the glutamate dehydrogenase. Okay. All right. Right. Yeah. Right. And now we come. So we understood ammonia, extrahepatic ammonia. How are that traveling? We understood glutamate in the liver, how it is that becoming alpha, basically the reverse reaction, alpha-ketoglutarate, and any amino acid entering the liver. All of these is taking in place in liver, except for this. This becomes liver here. Okay, so anything in taking, both of these are taking place in liver, and your ultimate goal here is to get glutamate here. And the glutamate further, again, as we know, gives back alpha ketoglutarate, so that is giving your ammonium back. All this is done. Now, there was one more shuttle that we had seen here, alanine. So let's look into this. Where does alanine come from? So alanine is basically, again, playing an important role in transporting amino group to the liver. And again, non-toxic form. Again, it has to come from muscle to liver, from brain to liver as glutamine. From muscle to liver, it will come as alanine. That's the shuttle we are talking about. So in muscles or certain tissues where you have degradation of amino acids for fuel, these amino groups, where you have degradation of amino acids, you have these amino groups, these are collected in the form of glutamate by transamination. Here you see the ammonium, you have glutamate, right? Now, glutamate can transfer its alpha amino group to pyruvate, and this pyruvate can convert to alanine, right? So this when glutamate gives its amino group to pyruvate, it becomes alanine, and it becomes alpha ketoglutarate, right? Now this is happening by alanine aminotransferase. Look at the name again, alanine aminotransferase. So it's basically transporting the amino group from glutamate to pyruvate, right? This alanine passes from the blood and travels to liver, and in the liver cytosol, again the same amino acid alanine converts the alpha ketoglutarate to glutamate. In case you haven't noticed, I had mentioned this here. Any aminotransferase has substrate of alpha ketoglutarate in liver. The moment alanine enters, it's basically this amino acid. This amino acid gets converted to its keto acid group, which is pyruvate, but subsequently using alpha ketoglutarate to give glutamate. That is exactly what is happening. So you have alanine. So in the blood, it is transported as alanine. Here we looked into in the blood, it's traveling as glutamine, right? So that's what is mentioned here. This glutamate ultimately will, as we know, will enter mitochondria of liver, and there it will uh, undergo the glutamate dehydrogenase to release ammonium, or can go transamination with auxiliary acetate to form aspartate. Is basically other groups that can take place. Now, what happens with the pyruvate in the liver? Where will it go? We know that pyruvate is converted to glucose for gluconeogenesis. It will go as blood glucose back. So this is a cycle which is making use of different metabolites, connecting each of them for different purposes. You have gluconeogenesis here, you have ammonium removal here, you have all these different song and dance going on for several of the metabolites coming together. But you need to remember, in amino acid catabolism, we need to come to this glutamate here. This glutamate is what will ultimately become alpha ketoglutarate, as we have just looked into by usage of glutamate dehydrogenase, and this ammonium will then be excreted. Now we have talked so much about ammonium. Let's look into urea cycle, what exactly is happening. So urea cycle is basically where you have these urea that you have just, or ammonium that you have just formed. It has to be excreted out, and that will do by the process of urea cycle, which will remove the, sorry, sorry, I thought there was a question. Yeah, this will pass into urine and be removed out of the cell. Okay, because remember, ammonium is toxic to the cell. Now urea cycle will begin again in the mitochondria. So what you need to remember is where in the liver it is taking place. It is taking place in the mitochondria, right? But before it's actually taking place in the mitochondria, there are few steps which is taking place outside in the cytosol. So that's why when you look into the urea cycle, you will see mitochondria and you will see cytosol. Remember, it's basically one single cell. And here you are, so let me just draw it out, maybe it becomes easier. Here is a cell, here is the mitochondria, here is a cytosol. So cytosol also has some steps taking place, and then it enters mitochondria where the remaining steps takes place. So that's why when you look into the urea cycle, you will find both mitochondria and cytosol. Okay, the beginning few reactions of there are some steps in cytosol and then there are some steps in mitochondria. So basically your urea cycle is present in both cytosol and your mitochondria. Okay. No, the first amino group to enter the urea cycle is derived from ammonia in the mitochondria. So you start with mitochondrial matrix ammonium, which we have so far seen that it arises for different pathways. And then liver will also receive ammonia via several others. So basically liver has ammonium in the mitochondria. Now this ammonium, which is generated in the liver mitochondria, it is, what is the first step? The first step is that it will combine with bicarbonate. Bicarbonate, HCO3 basically. So it will combine with HCO3, make use of ATP and get converted to something called carbamoyl phosphate. The enzyme here is, as you can see here, the enzyme will be carbamoyl phosphate synthetase. Again, synthetase, making use of ATP. Okay. So if you see here, so your this carbamoyl phosphate is made in the matrix. It's the ATP dependent step. Now the mitochondrial form of this enzyme is again, so there are two forms of this enzyme as well. In the carbamoyl phosphate synthetase, you have one in cytosol and one is in the mitochondria, right? So this carbamoyl phosphate, which you will find in the mitochondrial, this now functions as activated carbamoyl group donor, and this will enter urea cycle. So where does the urea cycle actually begin? The urea cycle begins when you have the carbamoyl phosphate, which is in the mitochondria. Where is it? Where you can locate? You locate it here. So all this glutamate is there, all the ammonium is there. Now the first thing ammonium will do is combine with SCO3 bicarbonate, use ATP, enzyme facilitating is carbamoyl synthetase, synthesizing carbamoyl phosphate using ATP, and a form of the enzyme that is in mitochondria is CPS1. Then you have this carbamoyl phosphate that enters the cycle, which is incorporated in the urea cycle. The first step is ornithine getting converted to citrulline. So this ornithine getting converted to citrulline is making use of the carbamoyl phosphate, and this is how your urea is getting incorporated. Don't get confused seeing these all these terms. We have done similar kind of activity in Krebs cycle also. If you remember, coenzyme A entering in the first step with the use of oxaloacetate. That's exactly what's happening here. You have this carbamoyl phosphate, you have ornithine. Both of these come together to form citrulline, right? Now, I will skip the details of which enzyme does what, but then just look overall. You have citrulline, which gets converted in two steps to arginosuccinate, which gets converted to arginine and fumarate, which gets converted to ornithine. Now, arginine, fumarate, arginosuccinate, everything that you see here are something that we have read here or there somewhere else. Again, in this step, you should remember citrulline getting converted to arginosuccinate again makes use of ATP. So this also is a step where you have, or process where you have multiple uses of ATP. But well, that's not important right now. What I want to bring your focus to is this step, last step here, arginine to ornithine, which is in the cytosol, is where you have the urea generation, and this urea is then transferred or transported and excreted out. The urea cycle, you should remember that it spans both mitochondria and cytosol, begins at mitochondria. Before the cycle, the first step is carbamoyl phosphate, and that carbamoyl phosphate is integrated into the cycle. Okay, right. And of course, if you know, so far as we have been looking into, all these steps are important, and each of these steps, they are regulated, and each of the steps are interconnected. See, this is mitochondria, this is a cytosol. Your majority of the steps of urea cycle is in cytosol, but you have this one step here which is in mitochondria. Now here you have citrulline. In mitochondria, you also have citric acid cycle, you have aspartate, you have citrulline. All of these are interconnected with each other. So what the whole agenda, what the whole intention here is that all the individual pathways that we have studied so far, we all should appreciate that they are all interconnected in some way, and all the intermediate, all the precursor or whatever metabolite you are seeing, they all are not just one specific for one specific pathway. They have use of several pathways, and that's what we are trying to connect here, right? Okay, I have spoken for way too long today, and I have also several questions today. I don't know how much I will be able to solve. What I will do is, let's say we are not able to, in case we are not able to complete all the questions are very diverse. Uh, People can drop off from the class in case it's too late, but my recording will be on, and I will show you all the questions, so that when you are looking back into the recording, or when you are looking into the uploaded video, you have all the questions there for your practice. The questions are all catered to the three topics that we have read: urea cycle, amino acid catabolism, and nitrogen fixation. So I have a lot of questions, and I hopefully I should be able to cover. They are not very difficult questions, but just in case we are not able to complete, I will, in the recording you will have all the questions there, right? Okay, on that note, let's begin with today's question answer session. So in case you have any doubt on any of the topics, let me know. I can go back to the slide and discuss. But for now, let's begin with the question answers, right? All these questions, you will see there is no particular separation based on the topics that we have read. I've purposefully combined all of these questions and given as a single topic because they are all both interconnected. And this will also give you a good revision of this topic. Okay, so let's begin with the first question. So you have different microorganisms here, bacteria, and you need to tell me which ones are incorrectly matched for their functionality. So you have several bacteria here. So Azotobacter, and it says it converts atmospheric nitrogen to ammonia. So it's basically about nitrogen fixation. And as I told you, you have these several steps here and different microorganisms in those steps. So it's a question from that part. So you have Nitrosomonas, Nitrobacter, Micrococcus, denitrificans, Klebsiella, and Rhizobium. And you need to tell me which one is incorrect. Which ones are incorrect? Maybe. B, C. That's it. Okay. All right. So the answer is B, C, D, and D. Okay. So let's look into this. Azotobacter is basically converting nitrogen to ammonia, which is, let me go back so that it becomes easier for you to understand. Yeah. So we have Azotobacter, which is basically converting the atmospheric nitrogen to ammonia, right? The second one was Nitrosomonas, and the option given was converting nitrite to nitrate. So nitrosomonas is here. It is incorrect. B is incorrect because it converts ammonium to nitrate, right? Third was Nitrobacter, which was saying that it converts ammonia in soil to nitrite, not directly ammonia to nitrate. It's nitrate to nitrate. That's why it's very incorrect. Fourth given was Micrococcus denitrificans, and it said that it converted nitrite into nitrogen. Incorrect. It converts nitrate into nitrogen. Okay, so that's why it was incorrect. Next was given Klebsiella, which converts nitrogen to ammonium. That's why it was correct. And the last one was again Rhizobium, which is for nitrogen fixation. That's part of the overall reaction. Yeah, BC, D also. I guess both of you have given the correct answer except for D. Denitrification, I understand where the confusion could be. Nitrite and nitrate, you need to be clear. One is NO3 minus, one is NO2 minus. And you need to know which one is getting converted. It's not nitrite which is getting converted. It is nitrate which is getting converted. The question says nitrite. Okay, might be thing, but you need to be very clear because nitrite and nitrate makes it two different components. Okay. Yeah. Cool. Okay. Let's do the next one. All right. Same again. I think it's nitrogen fixing. So it says there are 5 statements here. Read it out and let me know which one's correct. Okay. C, D, C, D, E. Okay, that's it. The question is about true. Okay, the answer is C, D, and E. So these are true statements. E as well. Yeah. Okay, we'll see why. Okay, let's look at the wrong sentences first. The first one says nitrogen fixing bacteria like rhizobium are present in the nucleus of the plant cells of the root nodule. No, nothing like that. It doesn't make sense. It basically is present in root nodules. Root nodules cells. These are cells on its own. There is no nucleus. There is no nucleus of plant cells involved there. Okay, and these are symbionts. So they are present in this special compartment. Okay. Next one says the reduction of nitrogen to ammonia is highly exergonic due to low activation energy requirement. No, we have established this extensively, right? That it is not correct because even though it has delta G as negative, your N2 bond is highly stable and you need a very high activation energy to overcome that, right? Okay. Third one says, which is correct, the activation energy of the reduction of nitrogen to ammonia can be attributed to the bond energy. That's exactly what we have established, right? Fourth one says ATP binding and hydrolysis at the dinitrogenase component of nitrogenase complex provide the energy required to overcome the energy barrier. Let me go back to that slide and show you once again. Right? So ATP is used here. Wait a minute. Yes. So dinitrogen component is using ATP. So you might be telling me it's nitrogen reductase. That's okay, but it means is the component as a whole is being utilizing ATP hydrolysis. And lastly, 6 electrons are directly involved in the reduction of nitrogen and ammonia. When you will look into the overall reaction, you will see that 6 electrons are involved when you are reducing N2 to NH3. So you will see stoichiometrically when you are looking into the equation, there will be 6 electrons that are directly involved in this conversion. Okay, we looked into 8 electrons. The 2 additional electrons are for the electron transfer system of the peroxide and the nitrogenous complex. But directly, the 6 electrons are being utilized to convert the nitrogen to ammonia. Right, and it says direct involvement. Okay, all right. Next. Yeah, this is from the nitrogenous complex. So this is about the two components, nitrogenase, the dinitrogenase and dinitrogenase reductase. So let me read it and let me know which one's correct and which one's not correct. B and E? Is that anyone else? Okay, no issues. While I am giving you the answer, you can check if you were thinking in the correct direction. So we are talking about which one's false, okay? And the answer, as you can see here, is B and E. I am, wait a second. D and E is something that we are saying is false. Uh, D and E. All right, we'll see. Okay, the first one is true, as we have already established that nitrogen complex consists of two enzymes, nitrogenous reductase and dinitrogen. That is correct. Now, B says dinitrogenous reductase is a heterodimer consisting of 4Fe, 4S redox center. Now, when you look into the structure, if you look into the notes, you will see a line saying the dinitrogenase reductase, that is what the question is about. So the dinitrogenase reductase is a dimer of two identical subunits. Wait, I'll come to this. Just give me a second. I will just come one by one. Maybe we will go over that. Sorry. Yeah, I'll come to the E question. I have seen that and I'll come back to it. But let's just first look into the B. B is incorrect because B is the correct statement will be dinitrogen reductase is a homodimer consisting of 4Fe, 4S redox center. Next one says P cluster consists of two bridge pairs of 4Fs, 4FeS redox center, which is correct. D says overall the nitrogenous complex is made up of 8 polypeptide chain. And did you say this is incorrect? Yes, you did say this is incorrect. Well, again, going back to your notes, you will see that dinitrogenous reductase is a dimer and dinitrogenase is a tetramer. So in no way you will see that it consists of 8 polypeptide chains. Okay. Okay. The last one says one cycle of electron transfer from the reductase to dinitrogenase requires the hydrolysis of 2 ATP molecule. Now, what did we say? We are saying it's wrong, but what did you say in the... Okay. E is correct because 16 by 8 is 2. 16 ATP by A, this thing is 2. Okay, yeah, I understand your logic, which says because we have established here that there is 16 ATPs, so you have use of 2 ATP molecules in one electron transfer. Well, yes, that's correct, but I will just look it up once again and then we can discuss. Yeah, I understand. Yeah, you're correct. I understand that for one electron there is 2 ATP. That's correct. They will also establish there are 6 electrons involved, and overall there are 8 electrons, so there are 16 electrons. Yes, I understand you're saying what you're saying is correct. Okay, give me 1 minute. I will come back to this question. In the meanwhile, you can try to solve this one. Give me 1 minute. I'll come back to the previous question. In the meanwhile, please solve this question, which says which enzyme catalyzes the oxidative deamination of glutamate? Okay, just give me 1 second. E is correct. Wait, just a second. So C, right? Yeah, so this answer is correct, as we have already seen. When you have deamination from glutamate, you have glutamate dehydrogenase. Coming back to it, okay, I think I also got confused when you wrote. Uh, what we are trying to highlight is which statement is false. So, statement is correct, right? It's a correct statement, right? So, the question here says which of the statements are false. So D is false and B is false. That means A, C, E are correct statements. That's why it was not highlighted. Yeah. I got confused thinking because I was following the same logic. All right. No problem. Once again, if any confusion was there, what we are looking into here is false statements. And the fifth one said one cycle of electron transfer from the reductase to dinitrogenase requires the hydrolysis of two molecules, one electron. Remember, we had 8 electrons and 16 ATPs. So if you follow that logic, you have 2 ATPs per electron. Okay, per electron transfer. So it was correct. All right. Yeah, and this question also we have established. Let's look into the next question. Which of the following enzymes participate in nitrogen disposal from the amino acid catabolism? Look into the name of the enzymes and see if they have a role in amino acid catabolism. Did we encounter these enzymes in the discussion today? B. Okay, that's it. Is it only B? A. That's it. Is that all? Okay. All right. Uh, okay. Uh, I don't know. Yeah, sorry. So the answer are A, B, C. Out of which I think you gave the answer as A and B, which was correct. So let's look into this. The first enzyme is carbamoyl phosphate synthetase. That was the probably the last enzyme that we looked into. It is part of the urea cycle, the first step of urea cycle where your ammonia is converted into a carbamoyl phosphate. So it is part of amino acid catabolism. Glutamate dehydrogenase, and we have just discussed, it's part of oxidative deamination of glutamate to give back the ammonia. Third, glutaminase. If you remember glutamin, which was how the toxic ammonia was transported in blood by converting ammonium to glutamin. This glutamin, when it comes to liver, it gets converted to glutamate, and that is because of glutaminase. So glutaminase is also part of amino acid catabolism. And succinate dehydrogenase is the incorrect option, because if you look here, let me go back to this shuttle here. Succinate dehydrogenase is basically here in the formation of fumarate, and it has no role with the urea cycle or any amino acid catabolism. So it has role in Krebs cycle, but it doesn't have a role in amino acid catabolism. All right, next one. Which of the following is the first reaction of urea cycle? We have just discussed it, so this is not a difficult thing to look into. Okay. Anyone else? Correct. Formation of citrulline, which is not in the option. So that's why the answer is none of the above. So you're basically saying D. D. Everyone is saying D. Well, the answer is correct for all of you. It's of course none of the above. But is the first step of urea cycle formation of citrulline or something else? Let me go back to the urea cycle for you to visualize it. Yeah. Which is the first step of urea cycle? Yes. Precisely this one. Not glutamate. Glutamate is where glutamate was part of the amino acid oxidation, right? When glutamate was formed from the glutamate, we have this step which is giving away of the ammonium. We have established that. When you talk about urea cycle, you're talking about ammonium. But that ammonium directly doesn't enter the urea cycle. The first step is where the urea is converted to carbamyl phosphate with the bicarbonate. Remember, that is the first step, not glutamate. When we say first step of urea cycle, the first step is this cycle, this step here, ammonia with HCO3 giving you carbamyl phosphate. Okay, and that enters citrulline, which was the second step. Someone wrote citrulline, right? Yeah, Neha. So Neha, the citrulline is there where it gets committed to the what do you say, uh, urea cycle, where it enters into the urea cycle. There's incorporation of carbamyl phosphate. But the first step of the cycle in itself is the conversion or the formation of carbamyl phosphate. Here, okay, I am moving ahead. If you have any doubt, post it in the chat box. All right, the next one. So in the glucose alanine cycle, which is the muscle cycle that we have looked into, muscle shuttle, alanine is transaminated in the liver to form which key intermediate of gluconeogenesis. So what happens when your alanine is entering in the liver again? What reaction is taking place and what is forming? Which intermediate form when you have? Okay. Anyone else? Pyruvate. C. C is alpha ketoglutarate. Okay. B. Pyruvate. So it says when alanine is transaminated, what is the key intermediate of gluconeogenesis, right? That's pyruvate. There are multiple key terms here that you have to identify the answer. One is transamination of alanine, and second is gluconeogenesis. Let's go back to the figure instead of me trying to make you imagine. Let's go here. So when it is transamination of alanine, your pyruvate is formed, and from alpha ketoglutarate, you have glutamate formation. Remember, there are two products, glutamate. That's when your alpha ketoglutarate is transaminated, and alanine is converted to pyruvate. That was the first clue. The second was it is part of gluconeogenesis. So pyruvate is part of gluconeogenesis. Glutamate undergoes the other pathways or other transformations. But when you have gluconeogenesis, it's basically talking about pyruvate, right? Okay. So the answer was pyruvate. All right. Next one. Which of the following statements about urea cycle regulation is true? Read it properly, read it carefully, and then try to answer. This is something that we didn't discuss in the text in theory, but that's okay. I wanted additional information when we are discussing the questions, so you can give it an attempt, and then we can look into the answers. C. Okay. Others? Yes, yes, I will. I told you I haven't discussed, so it's okay. I will just waiting for the answer. C, A, and D. All right. So let me give you the answers. The answers is C, A, yes, but not D, rather B. Okay, we'll do it one by one. It's not. So this is part of the regulation of urea cycle, which I did not do. So if you look into the theory, you will find part of which talks about urea cycle regulation. If not, it's easy to understand from here. Let's look into the first statement. It says N-acetyl glutamate, it activates carbamoyl phosphatase synthetase 1. And we know that carbamoyl phosphate synthetase 1 is the enzyme that is responsible for converting a formation of carbamoyl phosphate synthase. So N-acetyl glutamate, which is also called NAG, it is actually a truly activator of this CPS1, and this is the first step of the enzyme or first step of the urea cycle, right? And what it does is that how does it activate? It basically increases the enzyme's activity, and carbamoyl phosphate synthesis uses the ammonium and the bicarbonate to uh, form carbamoyl phosphate synthesis. So this is an activator, NAG. Okay. Next statement says glutamate and acetyl coenzyme are precursor for N-acetyl glutamate synthesis. So we are talking about the creation or the formation of acetyl glutamate, and it's in the name as only. Acetyl comes from acetyl coenzyme A, glutamate is from glutamate. So they both are required to form an acetyl glutamate synthesis. Someone did write B. Did someone write B? Okay. So B is correct. That's why B is correct. All right. The next one says the urea cycle is upregulated during prolonged fasting. This is easy to understand because if you are fasting, if you are starving, remember we have discussed, if we are in fasting or we are in starving, we have more ammonia production because protein is currently being used for fuel or for energy. And if your protein breakdown is taking place, you have more ammonia. And if you have more ammonia, you have your urea cycle in active form, right? And you need your ammonia right now because you are breaking up. So your urea cycle is upregulated to dispose the excess nitrogen that will be formed during this breakdown. So that's why it's upregulated, right? C was also correct. And so C someone mentioned, yes, C also someone mentioned. D says that aspartate availability does not influence. So it's a negative statement and it's incorrect because aspartate is important. I'll tell you where it is important. Aspartate gives you creates or forms the arginosuccinate. You have seen arginosuccinate here. Wait. here. And here you have aspartate. So you need aspartate and citrulline to form arginosuccinate. So you need aspartate in urea cycle. That's why this statement which says aspartate does not influence is incorrect. Right? Okay? You have any problem? Okay. If you have, as always, put in the chat box. I'll come back. Right. Let's do the next question. Right. Carbamoyl phosphate again. So you will see there are a lot of repeated questions. It's only the more you solve it in a question answer form, the more it makes it becomes more clearer and you get a hold of this topic. So carbamoyl phosphate donates its phosphate group to which compound to form citrulline? Why does it write catrulline? I don't know why did it write catrulline. It's citrulline. What's the compound which makes citrulline? B. Okay. Others? It's, I don't know why it says catrulline, but it's citrulline. So you have carbamoyl phosphate, citrulline, which is the precursor, which is the other product compound here. B. B, right? Ornithine. Let me go back again without any talking about or roundabout. Ornithine gives you citrulline with carbamoyl phosphate, right? That was something that we had discussed. Right. Next. Right. The regulation of glutamine synthase, we have discussed this topic, glutamine synthetase, involves which of the following mechanisms? So do you have covalent modification by adenylylation? Do you have allosteric inhibition by alpha ketoglutarate? And do you have feedback inhibition by glutamine? And do you have competitive inhibition by NAD+? In the previous one, I'm sorry. Which question, Neha? In the option C, please explain again. Neha, these glutamine synthase question or the phosphate group synthase phosphate group question? During fasting, that is oh, this one. Okay, I will come back to this question. We'll just finish this question first and then I'll come back. Okay, just because I've started this question, let me give the answer for this and then I'll discuss that. Cool. Okay, so let's first quickly do this and then we will go back to that question. So someone gave answer A. Anyone else in this question? Glutamine synthetase, which of these is responsible for regulation? Okay, no problem. I will give the answer for this and then we can discuss. Okay, the first one is the correct answer here and we know this, right? We have already seen this. Let me go back to the slide which showed covalent modification. Here, glutamine synthase, that was the question. And in glutamine synthase, it says how do we regulate. So the first option which says covalent modification by adenylation, it is definitely part of it, right? The second option said allosteric inhibition by alpha ketoglutarate. You don't see alpha ketoglutarate here. So alpha ketoglutarate is not responsible for inhibition. Next one said feedback inhibition activation by glutamine. Glutamine has, these are, I told you, these are all feedback inhibitions. So it says feedback activation, which is incorrect. All it does is feedback inhibition, that too by several intermediates, right? And the last one said competitive inhibition by NAD plus. Again, you can see there is no allosteric effect of NAD plus. So that's why the correct answer to this question is only A, which said that sorry, let it go. Covalent modification by adenylation. All right, this answer is clear. Now coming back to Neha's question, which was about yeah, this one, right, Neha? Yeah. All right. So here the question is, so under what conditions is your urea cycle regulated? And okay, and in this we are saying urea cycle is upregulated during prolonged fasting. We have discussed this. I have already shown this during the first slide here while we were discussing the topic. During starvations or in uncontrolled diabetes, when carbohydrates are unavailable or utilized properly, cellular protein are used as fuel. So the more cellular proteins are now being used in starvation, proteins degraded will form amino acid. Amino acid degraded will form ammonium. And we have established the fact that yes, of course, breakdown of protein, breakdown of amino acid is giving you energy, yes, but it is also generating lots of ammonium. Ammonium is toxic to our tissues, our cells. We know that. And ammonia needs to be excreted. So more the protein where will be degraded, more the amino acid that will be degraded, we know that more ammonia will be generated. So if you want ammonia to be excreted, you need to upregulate the urea cycle so that more of the urea is incorporated into the cycle and excreted out. Basically, more of the ammonia is incorporated in the urea cycle and more of the urea is produced so that it is excreted out, so that there is no ammonium toxicity in the cell. Does that make sense? Is it clear? So basically, you are fasting, you are starving yourself, you are not giving enough carbohydrate to yourself. Cell will start make use of different fuel that is already available there, right? One of them alternate source is protein. Remember? So protein is being used, protein which is already there, which is now being used as a fuel, which is stored. Now this protein is broken, amino acid is formed, amino acid is broken, ammonium is formed. And if you have ammonia, which is generating now in excess, this ammonia needs to be removed from the body. So you have to upregulate the urea cycle so that the ammonium is forming more of the urea and it is excreted out of the cell. It's it's this logic is that logical and it's that simple to understand. But is it clear, Neha?
Yes, ma'am.
All right, thank you so much. Okay, like I said that we are almost 2 minutes remaining for this session, so I will do one more question with you people, and then if you want to leave, you can leave. I will stay back and discuss all the remaining questions that are there just for you, so that so for you to know, like when you look into the recording which will be uploaded, as you know, you can look into those questions and solve it on yourself, because those questions are again something that will help you grasp the topic more. Right, we'll do one more question first together, and then if you have other work you can all leave. Okay. All right. The last question so far will is this. This is pretty straightforward, shouldn't take much time. So you are to give me the correct statements. B, C, D. Okay. E. Okay. B, C, D, E. Okay. All right. I will give you the answers because it's already 8. So if you want to leave. Right. So the answer is B, C, D. Okay. Someone said E. I will come to it. Why I didn't see E because I wanted to draw attention to something. But E, otherwise, it's also correct. Now the first one, cells can store amino acid obtained from diet in the Golgi. Cells cannot store amino acid. That's it. No Golgi, nothing. We have already said this that the amino acid are not stored in Golgi apparatus, right? They are stored as proteins, but amino acids are not stored. Okay, they are stored as proteins, so they are conjugated with each other and they form protein and those are stored. Now, next one said excess free amino acids in the cytoplasm can disturb osmotic balance. Yes, because amino acids they potentially can lead to cell swelling and disruption of cellular function. So that's why you don't have free amino acids roaming around here and there. You have amino acid complexed as protein, or they are bound as proteins. Like they bound to each other and form protein. Next one says the amino group from amino acid is converted into ammonia. We have already discussed this multiple times. Next one says the keto acid produced from aspartic acid is used in gluconeogenesis, and we have also looked into this. Next, so what does it form? It forms oxaloacetate. Okay, so if you have aspartic acid, the keto acid of aspartic acid is oxaloacetate, and we know oxaloacetate is important for gluconeogenesis. Now, the last one, or second last one says ammonia produced in the liver is combined with carbonic acid to form carbonyl phosphate. Carbonic acid is correct. When I was discussing, I was saying bicarbonate ions, so HCO3 minus is what I was trying to refer to. But if you look into HCO3, it comes from H2CO3. So so carbonic acid. So carbonic acid in itself is also correct answer. It's not incorrect. I just wanted to draw your attention to the fact that because we have been discussing about bicarbonate, if you stick to the terminology of bicarbonate, it is incorrect because we are talking about carbonic acid. But if you actually look into the logic, where will the bicarbonate ions come from? They will come from carbonic acid. So that is also correct. So both of these make sense. I wanted to draw your attention to this particular point. That's why I marked this as incorrect. But I definitely expected someone to give me this as a correct answer, and that's also correct. Okay. Last one says in human nitrogen is primarily excreted in uric acid. No, we have established that's birds. They are uricotelic. We are ureotelic. We excrete as urea, and fishes excrete as ammonia. All right. Okay. Well, here again, I would like to highlight that our class timings is over. So, in case you want to drop out, you can drop out. But I have some 10 questions more. Of course, if I discuss in regular class timing, it will take a lot of time. But if you wish to stay back, you're most welcome to. We can discuss this question. Otherwise, these questions will be available as recording. Right? All right. I will be continuing with the questions. If you people wish to stay back, you're most welcome to. Otherwise, we meet each other in the next session. No? Okay. Right. Uh, Coming to the next one, it says a mutation causes nitrogenase to have lower affinity for ATP. So if your nitrogenase has lower affinity for ATP, which one of this will be a outcome for this mutation? Yes, sure. Yeah. Please feel free to drop out, like I said. Thank you so much for attending today's session. We will meet in the next session. Yeah. All right. So what I will do is the remaining questions, I will wait for maybe 10 seconds if you people are on the call. 10-15 seconds for you to look into the question, and then we'll discuss the answer quickly so that we are able to finish as quickly as possible. Right. So the question here says that you have a nitrogenase. Remember, nitrogenase complex that we had discussed when we are looking into nitrogen fixation. So now nitrogen fixation, we know that this entire step requires ATP. So by a mutation in the enzyme, that enzyme is not able to bind more to ATP efficiently like it was doing before. So what will happen? By logic, we know that if my affinity for a particular substrate has gone down, the entire cascade of my reactions will be affected. So all the products will be affected, right? So that's the question. If you have a mutation in ATP binding or ATP affinity of nitrogenase, what will be the expected outcome? I will wait 10 seconds and then we will look into the answers. In the meanwhile, please do feel free to look into the answers yourself or try to attempt it. Okay. So the answer will be reduced nitrogen reduction, right? The most logical answer. The activity of nitrogenase is to reduce nitrogen. If you are, and it that reduction of nitrogen requires ATP. If my main enzymes in affinity for ATP has gone down, my nitrogen will not be reduced, right? So that's incorrect. I mean, that's correct. What the remaining steps are all incorrect. That's because the second one says increase ammonium production. Ammonium will form if you have reduction of nitrogen. If there is no reduction of nitrogen, if there is less reduction of nitrogen, we will not have more ammonium, because more ammonium means more of the reduction is taking place, but we have already established reduction is reduced. Second one says accumulation of nitrogen gas in bacterial cells. We know nitrogen gas is in environment. From environment, nitrogen gas is fixed into ammonium. If your nitrogen reduction is not happening, nitrogen gas is still in the environment and not in the bacterial cells. Last one says increased tolerance of oxygen for the enzyme. And we have already established, like someone said, A, absolutely correct. We know, and we have already discussed this, nitrogen is very, very sensitive to oxygen. So if you have a mutation in nitrogen, which lowers the affinity for ATP, doesn't mean that it will change its tolerance to oxygen. That is completely different, and that will not be affected because of lower affinity of ATP, right? Okay, quickly moving to the next question. Right, aldimine, as we have discussed, it is part of PLP. So these are some statements. You can pause when you are looking into the video and you can try and solving this. There are 6 statements here. While I'm reading the statements, you can also read and try to answer this. So what I am looking for is with regard to transamination, which is reaction where PLP is a prosthetic group of aminotransferase. During that reaction, which of the statements are true? So the aldimine, which is the aldimine form of coenzyme is covalently linked to the enzyme and the leucine residue. The aldimine can undergo transamination. The aldimine form can undergo decarboxylation. A carbanion intermediate is formed during the reaction. A quinoid intermediate is formed during the reaction. The reaction involves one substrate and produces two products, right? A D. Okay. ADE. Okay. Okay. Let me first bring you to the answers. It's B, C, D, E. Okay. So let me take you to the slide. This is a slide that is of importance to this question. So the first one says albumin is yeah. So the albumin form of the coenzyme is covalently linked to a leucine residue. It is incorrect. It is lysine residue. That's why it's false. Your deduction is correct, but it's not leucine. It's lysine. Okay. Okay. The albumin can undergo transamination. Yes. The albumin can go decarboxylation. Yes. A carbanion is formed here which undergoes stabilization is also correct. Yes. So let me look at the answer as D. So you said D is correct, which is this statement is correct. Along with this, the other two is also correct. We have seen that your cis base, which is the albumin group of pyridyl phosphate, it has three possible outcomes. So one is transamination, one is racemization, and one is decarboxylation. So we had already discussed this. That's why we know that these are correct. Next statement said that quinoid intermediate is formed. This is the intermediate here, which is correct. And then uh, coming back, it said that it requires one substrate and it has two products, which is not always the case because you can see that yes, one substrate and it is, I mean, it is giving two products, yes, but it also has an amino acid involved and a keto acid involved. So if you go back here, it has an amino acid and a ketoglutarate. So it has basically two of these substrates and two of the products. So that's why it's incorrect. Yeah. I'll go back again to the question just for you to have a look into it. So this one says one substrate, and we have just seen that there were two substrate, amino acid and alpha ketoglutarate. That's why it is incorrect. But otherwise, these statements we have already established. This one is lysine, right? Okay, and this one now. Very easy question. It basically says that you have enzyme on your left side and product on its right. So CPS is carbamoyl phosphate, and the name it suggests what it is. Ornithine transcarbamylase. What is the next component which is formed after ornithine? Arginase and arginosuccinase. These are all intermediates that we have seen in either Krebs cycle, either in urea cycle, and we know these names so far. See? Correct. Absolutely correct. So carbamoyl phosphate synthetase, carbamoyl phosphate is a product. B is 3 ornithine transcarbamylase gives you citrulline. C arginase gives you fumarate and arginosuccinase gives you urea. If you look into the urea cycle, this all will make sense to us. Okay, cool. Thank you very much. Let's do the next question. So you have these following statements. You need to tell again. These are all related to the transamination reaction. Which of these is false? These are again very simple. This is not at all difficult. A. We are looking at false statements. Okay. A. Only A. Okay, let me give you the answers. Like someone mentioned, it's A. A is the only false statement. That's also something that we have already discussed. Aminotransperases, it's a statement says aminotransferase are functionally distinct from transaminase, and we have established they are not. They are the same enzyme. They are basically two names from the same enzyme. Rest all statements are correct, and we have already discussed this. The coenzyme involved in transamination reaction is PLP, and it is derived from vitamin B6. Sorry. Third one says the mechanism transamination reaction is a classic example ping pong mechanism. So when you look into the detail of how this transamination takes place via PLP, you will see this mechanism, ping pong mechanism, which is basically one substrate, then an intermediate, then the other substrate, then it comes up, right? So you will see it in more detail there, but yes, it is a ping pong mechanism. Next one says aminotransferase involve pyridoxal phosphate bound via albumin linkage. We have already established this. Next one says excess amino acid in myocyte muscle cells are ultimately converted to alanine. Yes, and alanine is brought to hepatocyte for further degradation. Absolutely correct. Last one says amide nitrogen again of excess glutamine is released as ammonium ion in the mitochondria by the enzyme glutaminase in intestine, liver, and kidney. So we have mainly discussed liver, but kidney, intestines are also sites where you have the ammonium removal from the amino acids, right? So all of these are correct except for the first one. Fantastic. Next one. So if a bacterium loses, again, listen, look into it carefully and then try to answer. If a bacterium loses the ability to produce leg hemoglobin, which is basically important for nitrogen fixation, what would be the expected consequence in root nodules? This too is very simple. It says A. Any other statement? Okay, while you are looking at the other statements, the sentence or the incorrect, sorry, the correct ones are A and D. In the first says increased oxygen sensitivity of nitrogen. If you remember, the entire leg hemoglobin is there for oxygen sensitivity. The leg hemoglobin is a solution to the oxygen sensitivity of the enzymes, right? Nitrogen complex. So if your leg hemoglobin is not produced, that means your oxygen is not bound to the hemoglobin anymore. So your oxygen is always available for the nitrogenase to be interacting with, and it therefore has more sensitivity. That's correct. Second and third, increased nitrogen fixation efficiency is incorrect because the entire purpose of nitrogenase complex is to fix nitrogen. If your nitrogen complex is getting deactivated or inactivated because of oxygen, then it cannot do nitrogen fixation. Decreased ATP synthesis is not correlated or is not connected with the nitrogen fixation step. The last one says reduction in availability of ammonia. Yes, if ammonia reduction is not taking place, nitrogen reduction is not taking place, you don't have ammonia for the plant. So that's why it's also correct. Next, a scientist discovers a novel enzyme that converts alpha ketoglutarate to glutamate. We have seen this enzyme multiple times. So you need to tell me which one of these is likely required for its reaction. D. Yeah, just a second. Okay. So the answer is B and C. And let me tell you where did I get it from. Yeah, so wait a minute. Sorry, just a second. Yeah, so we said alpha ketoglutarate to glutamate, and for that, remember it requires. So, the question is based on function. Which of the following is most likely required for its reaction? You definitely need ammonia, right? So we are asking question like which one is definitely important. So you definitely need an ammonia because it needs to convert from the alpha ketoglutarate to glutamate. The other thing that is important is PLP, yes, and I don't know why it went to the second option, but yes, PLP as we have already established. So the answer here is C and D and not B. Ammonia unit because that's the enzyme, that's the group that it will transfer from alpha ketoglutarate to glutamate, and it's not B, it's D as we know it's important for the activity of aminotransferase. Okay, right. So it's B and D. I will correct it when I upload it, but the answer here is let me make it clear here. This one is not correct. This one is correct. Okay. All right. Moving ahead. Which are the following conditions would most likely increase the rate of biological nitrogen fixation? C and D. Well, this CS, not D. A. D will inhibit it. If you already have more ammonia, you will not require more nitrogen fixation, right? It's only when you don't have. So ammonia is a. Remember, we have discussed that when you have a product, it can also inhibit a pathway. So it will not increase the rate of biological. It doesn't require it to increase anymore. Similarly, oxygen on the contrary will rather inhibit it, right? Intracellular ATP, yes, because more the ATP, you can use it. So you can increase the rate of biological nitrogen fixation. And more ferredoxin, reduced ferredoxin will again increase the rate of fixation because you need reduced ferredoxin. So more number of the components which are actually used in the pathway, if they are high in number, your rate of fixation increases, not ammonia. Ammonia is an end product. So it doesn't directly increase, should not increase. It's if it is increasing, it means more of the reaction is already taking place. So if your concentration high, if you know high ammonia is problematic, you will not, this will not increase biological nitrogen fixation, rather it will upregulate urea cycle, right? Okay, next one, not last. Yeah, simple questions again. So you have 7 statements here and you need to tell me which one's true. Okay. B, C, D. All right. C, D, yes, not B. E, E is the correct answer. Well, let's look into this. So we are saying that which of the statements are correct. The first one says in all organisms, ammonia enter into amino acid metabolism through glutamate synthase reaction. We have looked into this, that there are many other ways for ammonia to enter amino acid metabolism. In some organism, it enters via glutamate dehydrogenase also. We have also seen this enzyme in function. So we know that via that pathway also you have ammonia entering metabolism. So it's not all organisms. Second one says, very important, glutamate synthase is a transamination. Incorrect. It's not transamination. Glutamate synthase is a reductive amination. What does it do? It makes glutamate, it synthesizes glutamate from alpha ketoglutarate and ammonia. That's a reductive amination, not transamination. Okay. Next one says glutamine synthesis is regulated by covalent. We have established this. P2 protein associated with adenylyl transfer is regulated by uridyl transferase. We have established this. ATP and alpha ketoglutarate inhibit deuridization. Is basically removal of UMP. And when you look into the regulation, that also we have discussed. Wait a minute. Sorry, there are way too many slides today, but yeah. I'm sorry, where did it go? Sorry. Where did it go? Yeah. So when we look into this, we have discussed that your sorry, just a second. So your glutamine synthesis is regulated by adenylation. We have established this. The P2 protein, which is this, associated with adenylyl transfer, is regulated by uridyl transfer mediated uridylation. We know this. We have seen that ATP and alpha ketoglutarate. It says the statement was ATP and alpha ketoglutarate inhibit the deuridylation. And when you look into this, you will see that it's not correct. And inorganic phosphate and glutamine. So when you look into these statement, you can see glutamine and inorganic phosphate inhibit it. Okay. So you will see that these statements are incorrect. So we have already discussed this. So when you look into this figure, you will understand that the statements which were the just a second, there are too many statements. Yeah, these two statements are incorrect. Okay. All right. Next one. We have very few questions remaining. Yes, like I guess 3 more. Yep. Okay. So if aspartate aminotransferase is an enzyme, aminotransferase inhibited in liver, aspartate, you need to remember which one is the keto group, keto acid of aspartate. So if that is inhibited, which one of the following is affected? C. C is correct along with A. I told you remember this thing, keto acid, right? So your aspartate amino acid transferase actually is responsible for the conversion of aspartate, as we know, and alpha ketoglutarate. That also we know that every time there is an amino acid that is supposed to be transaminated, you have alpha ketoglutarate as a substrate. So that gets converted to glutamate, and your aspartate gets converted to oxaloacetate. So if your aspartate aminotransferase is not working or inhibited, you have decreased oxaloacetate availability because it's not formed only. Second one is increased nitrogen expression. Well, nitrogen expression is basically ammonia, and ammonia is formed when you have these enzymes, amino acids getting converted, and it's not getting converted here, so you cannot have increased expression. The third one says impaired gluconeogenesis. Yes, we know oxaloacetate has a role in gluconeogenesis. So if it is decreased, your gluconeogenesis is also impaired. And last one says transamination of alanine. Increased transamination is incorrect because it's not increasing anymore transamination. It's rather for transamination of alanine, it's not responsible. It's not directly playing a role, right? But it affects the other pathways which involves, which affects the transamination of alanine. So in any case, there will not be increase in the transamination. Right. I guess the second last question. It says which are the following enzymes participate in nitrogen assimilation in bacteria? Did we do this already? No. Yeah. A, B, and D. Sorry. Uh, A, B, definitely yes. Aspartate aminotransferase is not, right? It's for breaking that we know of. And nitrate reductase is actually basically for conversion from nitrate to nitrite. Uh, well, it is a part of nitrogen assimilation, but when I was so you can put it as an answer, but so I will consider that as an answer. So ABD is correct because your nitrate reductase is also basically used for conversion of nitrate to nitrite, which is not directly, but also they are part of the nitrogen fixation. And nitrogen fixation is what gives you ammonium, which further takes part in nitrogen assimilation. So yes, I will take D. But what and I was answering, I was pertaining directly to the conversion of ammonium. That's why I didn't put D. But if you look in the broader picture, then that is also correct. Yeah. And the last question for the day. Which one of the following are correct regarding the fate of nitrogen by symbiotic bacteria? Very, very easy. That's absolutely simple question. A, is that it? Okay, let me give you the answers. A is definitely correct, of course. If they are fixing nitrogen, they are converting into ammonia, which is assimilated into glutamine and glutamate. Second one is incorrect because, of course, why will they say store as nitrogen? They are basically helping it to convert to ammonia. This is fixing nitrogen, is reducing nitrogen. Third one says it supports plant growth. Yes, because it is the whole agenda here, right? Your ammonium, which is being produced, it gets incorporated. Remember nitrogen assimilation, so it is incorporated into amino acids such as glutamate. We have already discussed this, and this further give rise to protein synthesis, which is important for plant growth. So it is also incorporated into plant growth, and it is not excreted as nitric oxide. It is converted to ammonia, which is used for uh, nitrogen assimilation, right? Okay. Okay, well, that brings me to an end of today's class. Thank you so much for being with me, and we had this lot of questions discussed. And the next class, I think it's fatty acid part that we'll be covering. And like always, I'll try to put more questions so that we have this extensive discussion that makes you have a question bank that you can use for your revision. So that was the end for today's class, and thank you so much for being with me. If you have any doubt, let me know. Else, we can meet in the next session.
Thank you, madam. Thank you so
Thank
much.
you. Thank you so much.